Chapter 7. Genetic Engineering for Human Welfare. Following the recombinant DNA techniques, thousands of DNA fragments of prokaryotes and eukaryotes, plant and animals, have been cloned during the last 0 decades. According to the utility, the cloned DNAs can be used in basic research or commercial production of useful products. Basic research includes DNA probe, expression of cloned genes, colony plaque hybridization, genome mapping, and many more related to genetic engineering. Scientists have successfully cloned mammals and crop plants. The new areas of application of genetic engineering are now being explored. It has been used in medicine, pharmacology, reproductive technology, and several ether research areas. Obviously, genetic engineering holds the key in curing many serious diseases, which have claimed many lives. You know that the production of insulin was one of the foremost endeavors for human welfare. Nowadays, insulin is being produced through genetic engineering. Interferon secreted by human cells combats virus attack can also be produced commercially in recent years. Gene therapy is also one of the major benefits of genetic engineering, where diseases are cured by repairing or replacing the abnormal non-functional human genes or introducing therapeutic genes, such as Huntington's disease, cystic fibrosis, etc. Genetic engineering has also played a key role in the production of new pharmaceutical products. These new pharmaceuticals are created by cloning certain genes. The genetically manufactured human growth hormones are extremely effective in the treatment of skin burns, bone fractures, ulcers in the digestive tract, etc. Production of genetically modified crops (GMPs) has been possible through genetic engineering. Some of the most important GMO crops are corn, soybean, seed oil, cotton, etc. These crops are resistant to insects or deleterious herbicides. The field of genetic engineering is also involved in production of novel items such as blue roses and glowfish, etc. The scientists can use this tool for the welfare of the entire human race. Moreover, cloned genes are utilized commercially in medicals, agriculture and industry, and the production of valuable products. Some of the cloned genes and application of genetic engineering techniques are described here. A cloned genes and production of growth hormones, vaccines and commercial chemicals. 1. Human peptide hormone genes. In the human body, peptide hormones are secreted after encoding the peptide hormone genes in the specialized cells, for instance insulin and other human growth hormones, HGH. A. Insulin. This peptide hormone, i.e. insulin, is secreted by the islets of Langerhans of pancreas, which catabolizes glucose in blood. Insulin is a boon for the diabetics whose normal function for sugar metabolism generally fails. Insulin consists of two polypeptide chains, chain A 21 amino acid long and B 30 amino acid long. Its precursor is proinsulin, which also contains two polypeptide chains, A and B, and is connected with a third peptide chain C 35 amino acid long. However, the recent discoveries reveal that precursor of insulin is proinsulin, which is about 109 amino acids long. The proinsulin is synthesized in beta cells of pancreas, the structure of which is given below. NH2, peptide, sus chain, peptide C, A chain COOH. In the beginning, efforts were made to isolate mRNA for proinsulin from rats' islets of Langerhans of pancreas and to synthesize cDNA. Thereafter, it was inserted into a plasmid. The recombinant plasmids were transferred into the E. coli cells, which secreted proinsulin. K. Itakura and collaborators in 1977 chemically synthesized DNA sequence for two chains, A and B, of insulin and separately inserted into two PBR322 plasmids by the side of sus-galactosidase gene. The recombinant plasmids were separately transferred into E. coli cells, which secreted fused sus-galactosidase A chain and sus-galactosidase B chain separately. These chains were isolated by detaching from sus-galactosidase in pure form in an amount of about 10 mg, 24 grams of healthy and transformed cells. Production of recombinant insulin is shown in Figure 7.1. See Figure 7.1. Production of recombinant insulin in E. coli. Detachment of proinsulin could be possible when an extra methionine codon was added at the N-terminus of each gene for A-B chains. The two chains, A and B, were joined in vitro to reconstitute the native. Sulfonating the two peptides with sodium disulfonate and sodium sulfite. In 1980, W. Gilbin and L. Vilikomarov isolated mRNA for insulin from sus cells of rat's pancreas and inserted PBR322 plasmid in the middle of a gene normally coding the penicillinase, and into E. coli cells. E. coli cells produced a hybrid protein penicillinase proinsulin, from which the proinsulin was separated by using trypsin. It is estimated that clones of E. coli are capable of producing about 1 million molecules of insulin per bacterial cell. Human insulin, Homulin, is the first therapeutic product produced by means of recombinant technology by Eli Lilly and Company. In 1980, Shreya Life Sciences Pune, India company has started producing the second generation rDNA-based insulin without using DNBR with the name Ricosulin. B. Somatotropin. Somatotropin is secreted by the anterior lobe of pituitary glands, which consist of 191 amino acid units. Its secretion is regulated by two other hormones, somatostatin and growth hormone releasing hormone produced by hypothalamus. Deficiency of somatotropin in about 3% cases is of hereditary. It has been estimated to about a child in 5,000. Turner's syndrome is one of the most common chromosome disorders in girls, and it is characterized by short stature and non-functioning of ovaries affecting approximately I in 2,500 live female birth. The extraction of somatotropin pharmaceutically from the pituitary glands could not meet annual demand of this hormone. Biosynthesis of somatotropin was achieved through gene cloning procedures. Double-stranded cDNAs were produced from mRNA precursor of HGH gene, which was then incorporated into bacterial cells where it expressed in non-precursor form. Bacteria were unable to convert peptide into biologically active form. A recombinant plasmid containing a full-length HGH cDNA, which fails to express, is cleaved with restriction enzymes that release a fragment containing the complete HGH coding sequence after codon 24, figure 7.2. Several overlapping complementary oligonucleotides are ligated to form one synthetic strand of small DNA fragment which contains the coding sequence for the first 24 amino acids of macho HGH after removal of the N-terminal signal peptide. The synthetic DNA and cDNA molecules are ligated to yield a new fragment which contains the complete coding sequence of HGH DNA. It is ligated into a restriction site just downstream of the lac promoter operator region cloned on a plasmid. The rDNA plasmids are allowed to transform E. coli. Synthesis of HGH is induced by an inducer of lac operon, i.e. isopropyl thiogalactoside (IPTG). The HGH is subsequently purified. HGH is being produced commercially by this method. About 100,000 molecules of hormone per cell of E. coli have been produced. Newmark, 1979. One of the major difficulties was that at the end of polypeptide an extra methionine, the MET-HGH, was attached. Methods are developed to remove methionine from the MET-HGH molecules. C. Somatostatin. Somatostatin, a 14-residue polypeptide hormone, is synthesized in the hypothalamus. It is the first polypeptide which was expressed in E. coli as part of the fusion peptide which inhibited the secretion of growth hormone, glucagon and insulin. It does not contain any internal methionine. 8 single-stranded DNA segments were synthesized chemically which were annealed in an overlapping manner to form a double-stranded DNA, the synthetic gene. It had single-stranded projections at the each end as the same are formed by E. coli. The synthesized gene contained 51 base pairs which were terminated by two nonsense stop codon N preceded by a methoin codon as below. ATG, 42 base pairs encoding somatostatin GATAG. Two plasmids, PSOMI and PSOM2-3, were constructed. The synthetic gene was introduced into E. coli 3-galactosidase gene at different sites. See figure 7.3. Synthesis of somatostatin, LAC-PO, lactose promoter operator, ST, somatostatin, AMP, ampicillin resistance. The chemically synthesized gene was inserted downstream from the lac promoter in such a way that the gene fusion should have specified a polypeptide in which the first 7 amino acids of cis-galactosidase were fused to somatostatin. But the somatostatin was not detected in transformed bacteria, possibly it was degraded in coli. Glover, 1994. An alternative plasmid, PSOM11-3, was constructed which had the both EAC promoter operator and LUX regions. The LUX gene encodes peptide of cis-galactosidase. If resulting frame of LUX is maintained after inserting a DNA, a fusion peptide is produced. The plasmid was cleaved in the Lux segment by restriction enzyme figure 7.3. The synthetic gene was inserted into the plasmid at eco-re site near C-terminus of sus-galactosidase. The plasmid in the transformed bacterium directs the synthesis of a fused protein consisting of INH2-termined segment of sus-galactosidase fragment coupled by methionine to somatostatin. It was stabilized from proteolytic degradation by sus-galactosidase moiety, Glover, 1984. This fused protein was purified and treated with cyanogen bromide, CNBr, which cleaves only protein at the carboxy side of the methionine. Thus, the methionine linker remains attached to sus-galactosidase fragment and somatostatin was released. Now, it has become possible to inhibit the degradation of foreign protein in E. coli by introduction of protease inhibition PIN gene OFT4 phage. D. Sus-endorphin. Sus-endorphin 30 amino acid long neuropeptide with opiate activity is another growth hormone which was expressed in genetically engineered E. coli cells. In 1980, J. Shine and co-workers integrated DNA sequences of sus-endorphin, obtained from mRNA, adjacent to sus-galactosidase genes on plasmid. The mRNA contained large precursor of protein that consisted of, besides sus-endorphin, the hormones of melanotropin, corticotropin, sus-lipotropin and sus-melanotropin. The sus-endorphin is cleaved from C-terminus of the precursor peptide. In this way, the transformed bacteria produced an insoluble fusion protein between sus-galactosidase and sus-endorphin. Endorphin can be removed from the hybrid protein by trypsin, which cleaves only at arginine residue. Before doing so, internal lysines are protected from trypsinization by citraconylation as below. See the figure. 2. Human interferon genes Hig. For the first time, Isaacs and Lindenman isolated the interferon in 1957. Definition and nomenclature of interferon have been recommended by a committee of experts. Interferon is defined as a protein which exerts virus non-specific antiviral activity, at least in homologous cells through cellular metabolic procedure involving the synthesis of both RNA and protein. Thus, interferon is secreted by human cells just to resist the immediate invasion by virus and multiplication of abnormal cells. Interferon is used to cure many viral diseases such as common cold and hepatitis. It is species specific. In man there are three classes of interferon: 1. Alpha interferon IFNU or leukocyte interferon, leukocytes of blood; 2. Beta interferon IFN SUS of fibroblast interferon, fibroblast of connective tissue; 3. Gamma interferon IFN7 or immune interferon by lymphocytes of blood, and lymphoblastoid interferon by transformed leukocytes. According to origin of cells, they are classified into two major groups: leukocyte interferon and fibroblast interferon. Leukocyte interferon is produced on large scale, but major difficulty is that mass production of IFN cannot be done. In 1980, IFNA and IFN SUS was successfully produced from genetically engineered E. coli cells by isolation of mRNA from leukocytes and fibroblasts, production of cDNA, its integration into PBR322 and incorporation and cloning into E. coli cells. Production was estimated to be about 1,000 to 100,000 molecules of IFN SUS per cell. The Swedish firm Biogen produced and IFN SUS through recombinant DNA techniques. It was found that genes responsible for the production of IFNU and IFN SUS had 865 and 836 nucleotides, respectively. Later on, hybrid plasmid containing cDNA of IFN SUS genes was built up, which needed a promoter site on plasmid to express in E. coli cells. Similarly, hybrid plasmids were also prepared that contained IFNgens with trap promoter between the leader and ribosome binding sites, so that expression of interferon could be done. Expression of both the interferon could be optimized by varying the spacing sequence between trapped Schneider-Algernon sequence and the initiator codon. IFN SUS produced by genetically engineered microorganisms showed lower specific activity and decreased stability than natural one. Enhanced specific activity and stability was obtained when the cysteine at position 17 was replaced by a series of site-specific mutagenesis resulting in JFN SUS molecule. It was stable for 2 years and well-tolerant in cancer patients. Moreover, genetically engineered E. coli is reported to yield 5 to 10 million units per milliliter of IFN SUS in a 200-liter batch reactor within 2 to 3 days of fermentation. 3. Genes for vaccines and immunogenic substances. Vaccines are chemical substances prepared from the proteins, antigen, of other animals which confer immunity to a particular virus. Some of the vaccines synthesized biologically through genetic engineering are briefly described as below. A. Vaccines for hepatitis B virus. Hepatitis B virus (HBV) is widespread in man and produces several chronic liver disorders such as fulminant chronic hepatitis, cirrhosis, and primary liver cancer. HBV DNA is a double-stranded circular molecule of about 3 kilobits size and has a large single-stranded gap which must be required with an endogenous polymerase before digestion with restriction enzyme for DNA cloning. After infection in human being, HBV fails to multiply and infect a large number of cells and even does not grow in cultured cells. This property has been explained to be due to hindrance of its molecular characterization and development of vaccines. Plasma of human has been detected to have varying amount of antigens. 3 types of viral proteins are recognized to be antigenic. 1. Viral surface antigen HBsAg, 2. Viral core antigen HBcAg, and 3. The A antigen BIG. Although the whole viral genome has been cloned and sequenced, yet there is limited information about amino acid sequence of surface and core antigens. Recently, HBV DNA has been successfully cloned in E. coli in mammalian cells, and synthesis of HBsAg and HBcAg particles has been done in the cells. In 1979, C.J. Burrell and co-workers inserted HBcAg genes in PBR322 near Sus galactosidase gene. Production of these genes is needed in order to get production of vaccines on a large scale. In yeast or mammalian systems, these antigens are synthesized more efficiently than in prokaryotes. 1. Recombinant vaccine for hepatitis B virus. After infection, HBV fails to grow and even in cultured cells it does not grow. This property has been explained to be due to inhibition of its molecular expression and development of vaccines. Recombinant vaccine for HBV was produced by cloning HBsAg gene of the virus in yeast cells. The yeast system has its complex membrane and ability of secreting glycosylate protein. This have made it possible to build an autonomously replicating plasmid containing HBsAg gene near the yeast alcohol dehydrogenase ADI promoter figure 7.4. The HBsAg gene contains 6 BP long sequence preceding the August that synthesizes intermanol methionine. This is joined to AD promoter cloned in the yeast vector PMA56. The recombinant plasmid is inserted into yeast cells. The transformed yeast cells are multiplied in tryptophan-free medium. The transformed cells are selected. The cloned yeast cells are cultured for expression of HBsAg gene. This inserted gene sequence expresses and produces particles similar to the 22 gm particle of HBV as these particles are produced in serum of HBV patients. The expressed HBsAg particles have similarity in structure and immunogenicity with those isolated from HBV-infected cells of patients. Its high immunogenicity has made it possible to market the recombinant product as vaccine against HBV infection. 2. Indigenous hepatitis B vaccine. In India, first genetically engineered vaccine, Guni, against HBV was developed by a Hyderabad-based laboratory, Shanta Biotechnics Private Limited. India is the 4th country after the USA, France and Belgium to develop this highly advanced vaccine. The indigenous yeast-desired HBV vaccine is 1/3 the cost of the imported vaccine. This new vaccine had undergone human clinical trials at NISM's Institute of Medical Sciences, Hyderabad and KEM Hospital, Mumbai. The clinical trials clearly proved that the seroprotection is about 98%. It was found more effective than the imported vaccine. The Drug Controller General of India has permitted it for commercial manufacture. See the figure 7.4. Expression of HBsAg gene in yeast. B. Vaccines for rabies virus (RV). RV causes hydrophobia in animals and humans in many countries like South America, Africa, Asia, including India and Pakistan. Researches are being done to synthesize vaccines by inducing genetically engineered E. coli cells. However, attempt has been made to isolate mRNA encoding viral protein from RV-infected cells. The genes coding for the production of rabies virus glycoprotein coat has been successfully transferred to E. coli. This is the first step towards the production of antirabies virus vaccines as this glycoprotein stimulates antibody production in diseased animals. In 1984, Vista Institute in Philadelphia developed a new genetically engineered vaccinia virus by inserting a small piece of foreign DNA. The genetically engineered virus synthesized antirabies vaccines for animals. The recombinant vaccinia virus did not cause rabies in those animals that received rabies genome but encoded antigenic molecules and activated the immune system against rabies infection. This new vaccine can be administered orally in animals, and could be decreased the risk of human death due to bites of animals receiving rabies virus. C. Vaccines for poliovirus. Poliovirus is a causal agent of poliomyelitis in human beings. First attempt to describe the complete structure of poliovirus RNA was made by N. Kitamura and co-workers in 1981. Since then it has become an easy task to have the expression of genetic information of poliovirus. Anti-poliomyelitis vaccines through genetically engineered bacterial cells have not been synthesized so far. D. Vaccines for foot and mouth disease virus FMDV. Foot and mouth disease FMD is a serious disease caused by aphthovirus. The primary control measure of the disease has been the slaughter of FMDV infected animals. Chemotherapeutic way of FMD control is vaccination. Vaccines are produced by inactivation of virus grown in bovine tongue epithelium. A detailed study of FMDV reveals that it contains a single-stranded RNA covered in a capsid of 4 polypeptides, for example, VPL, VP2, VP3 and VP4, where only VPI has a little immunogenic activity. However, the nucleotide sequence encoding for VPI was identified on the single-stranded RNA genome and cloned on double-stranded PBR322 in E. coli. About 1,000 molecules of VPI per bacterial cell were synthesized. Vaccines produced from genetically engineered E. coli cells cannot compete with those extracted from virus particle. Therefore, much more work is to be done to make the vaccines available for FMDV on a large scale at low price. An outline for production of FMD vaccine is given in Figure 7.5. Synthetic peptides have also been produced and used for immunization against bacterial diphtheria, Streptococcus pyogenes, and viral hepatitis virus, FMDV, diseases. For the production of synthetic polypeptide to be used as vaccines, it is necessary to have the knowledge of structure and function of proteins in the regions involved in immunogenic response. For example, synthetic polypeptides having immunogenic effects against HBV contain disulfide bond in the region between amino acids 117 and 137 corresponding to the viral surface antigen. After injection into mice, the polypeptides elicited antibodies against HBsAg and protected half of mice. Similarly, a synthetic polypeptide has been identified that corresponds several regions of FMDV protein V place. The region between amino acids 141 and 160 elicited and production of antibodies against FMDV in guinea pigs, rabbits and swine. Likewise, another polypeptide was synthesized and the region between amino acids 200 to 213 VPI also elicited antibodies against FMDV. There are other examples also for the synthesis of vaccines against viral diseases. Such vaccines too have a great promise for the immunization purposes. A. Vaccines for smallpox virus. Smallpox is a very serious disease of humans in most of the countries. The vaccinia virus, the cowpox virus, can be used as the basis of the smallpox vaccine by using the recombinant DNA technology. The genome of vaccinia virus is altered by inserting the foreign genes which encodes the pathogenic antigens. When a person is immunized with a vaccine in preparations, the foreign genes enter the body cells of the patients receiving vaccinia virus and direct the synthesis of the foreign antigenic proteins. The genes replicate like vaccinia genes and induce immunity to the smallpox. However, it resembles the other of vaccine. The vaccinia virus can be modified in such a way that a single preparation confers protection against many viral disease. See Figure 7.5. Recombinant DNA technology for making foot and mouth disease vaccine. Malaria vaccines. According to WHO estimates 4 billion people are at the risk of developing malaria and about 500 million cases occur each year, resulting in 1 million deaths each year, mainly of children of 5 years age and pregnant women. In addition, development of resistance against drugs by the species of Plasmodium, and insecticides by mosquitoes have been reported. Therefore, threat of malaria disease is still increasing for humans. Therefore, for control of malaria, use of vaccines and vector control programs would be successful. Much work is going on at Indian Institute of Immunology, New Delhi, and ICGEB on development of malaria vaccine by using modern methodologies. All kind of vaccine development through recombinant antigens, synthetic peptides and direct use of DNA are being attempted. All these attempts indicate that development of malaria vaccine is largely complex process. However, progress towards the development of a malaria vaccine has been slow due to several, one of which has been the lack of in vitro correlates in the suitable animal models for malaria vaccine trials. Plasmodium rhesus monkey is one of the models for malaria vaccine development. Malaria vaccines are being developed at three distinct developmental stages of the parasite. 1. Pre-erythrocytic stage to eliminate infection by blocking the sporozoites from entering hepatocytes or by destroying the infected hepatocytes. 2. Blood stage of parasite to prevent disease or reduce parasitic load. And 3. Sexual stage parasite to limit transmission of disease. In India, 60-70% malaria is due to PYVX, which do not kill host but results discomfort and morbidity. It is more prevalent throughout Asia, but less is known about immune response of host against PYVX as it resisted all attempts of culturing the parasite. P. cynomolgi is a simian malaria which is closely related to PYVX in taxonomy and morphology. Hence, it is regarded as a good model to study PYVX infection as both share a similar clinical course of infection. At present, vaccines are being developed at ICGEB against all stages of life cycle of the parasite, but it is that an asexual blood stage vaccine is most likely to have the greatest impact on the disease. See Table 7.1 Asexual stages vaccine target antigens. 1. Expression of vaccine target antigens. The most successful vaccines have been based on attenuated or killed pathogens. Malaria vaccine is limited to well-defined molecules which can induce protective immune responses and easily produced by recombinant DNA technology in various systems. 3. Important vaccine target antigens such as thrombospondin-related adhesive protein trap, apical membrane antigen AMA, and erythrocyte binding protein EBP from P. cynomolgi have been cloned and sequenced. Table 7.1. To evaluate their vaccine potential, these antigens have been expressed in E. coli using PQE expression system. Each of these proteins was expressed at high levels as insoluble inclusion bodies. Protocols for the large-scale production of the correctly folded PC trap have been developed. Trap has a multidomain structure and localized on the cell surface of P. falciparum sporozoites. 2. Animal trials of malaria vaccines. The rhesus monkeys were immunized with recombinant PC trap parasite lysate or adjuvant to study the protective efficacy. The antigens were delivered intramuscularly in 3 doses 500 gg each, on 0, 42, and 62 days. Blood from immunized and unimmunized monkeys was collected on days L, 14, 29, 52 and 70. High antibody titers 8 to 16x105 and above were detected against PC trap and parasite lysate as measured by ELISA technique. Then immunized monkeys after injection with P. cynomolgi sporozoites 3x104 were protected from malaria. Merozoite surface protein MSP119, the cysteine-rich C-terminal domain of on the surface of R. falciparum, is a leading malaria vaccine candidate. This is the only part of protein which remains bound to the merozoite membrane after invasion. Similarly, the expression and purification of R. falciparum acidic basic repeat antigen ABRA and its fragments from E. coli has also done. The purified recombinant proteins have been used to assess the antibody responses in human populations living in malaria endemic areas from Kalka village of Rourkela, Arisa, and from Nigeria. It has also showed protective efficacy in immunized fabbits. SPF 66 was the first recognized DNA vaccine for malaria developed by joining 3 merozoite-derived proteins with repetitive sequences derived from the circumsporozoite protein of P. falciparum. This vaccine has given equivocal results on human trials in more than 1 locations. Through Indo-US collaboration, a recombinant multistage P. falciparum candidate vaccine has been developed. DNA vaccines. For the first time, J. A. Wolf and co-workers in 1990 injected naked DNA into the muscles of mice, which led to expression of encoded marker protein. Thereafter, there has been a search to use this approach to generate DNA vaccines against a variety of infectious diseases. Thus, DNA vaccines are giving hope of a third vaccine evolution. The first published report from India indicates modest success in the development of DNA vaccines against rabies and Japanese encephalitis virus (JEV) experimental animals. The efficacy of DNA vaccine G protein against rabies is correlated to levels of neutralizing antibodies, whereas in the case of JEV enveloped protein, cell-mediated immunity appears to be the major mechanism of protection. There is great hope that DNA vaccines can protect against serious infectious diseases. Most likely it is to become a to benefit mankind in 21st century as such or in combination with combining cell culture vaccines or as an adjunct to chemotherapy. In case of malaria, DNA vaccines have distinct advantage where plasmid DNA encoding different antigens and prepared by the same genetic procedure can be mixed and administered. A mixture of 4 plasmid DNA, PFCSP, PFSSP2, FEXPL and PFLSA1 has been injected into monkeys and found to elicit multiple antigen-specific cytotoxic T lymphocytes. D. H. Laurie and co-workers in 1999 reported that a DNA vaccine coding for a mycobacterial heat shock protein of Mr. 65,000 HSP65 when administered in 4 doses to mice, 8 weeks after intravenous injection of virulent M. tuberculosis E137RV, leads to a dramatic decrease in number of live bacteria in spleen and lungs 2 months and 5 months after the first dose of DNA. Certain other mycobacterial antigens and BCG did not have this effect. 4. Production of commercial chemicals. There are several chemicals which are produced by using the recombinant DNA technologies. A few of them are as below. A. Vitamins, see Chapter 17. B. Organic acids, see Chapter 17. C. Alcohols, see Chapter 17. D. Antibiotics, see Chapter 17. Recombinant DNA technology has helped in increased production of antibiotics. For example, the rate of penicillin produced at present is about 150,000 units per milliliter against about 10 units per milliliter in 1950s. However, protoplast fusion technology of microbes for antibiotic production holds promise for microbes increasing the rate of production. The hybrid cell manifests genetic features of both the species. The hybrid species can produce new antibiotic or increase the productivity of the strain. For example, hybrids obtained from the protoplast fusion of Streptomyces griseus and S. tanimatiensis produced a new metabolite of entirely different properties. B. Prevention, diagnosis and cure of disease. Prevention of many genetic disease in early period of pregnancy will help the mothers to give birth to the healthy babies.